in fact, first step in any type of structure prediction, and that is the isolation and purification. And we have been focusing, we are currently focusing on this isolation and purification stuff. And we have seen yesterday that there are multiple techniques which could be used to purify and isolate the proteins. One of those, one of the technique was centrifugation, जिसकी हमने बात की थी. So there were two types of centrifugation. एक सेंट्रीफ्यूजेशन डिफरेंशियल सेंट्रीफ्यूजेशन थी जिसके अंदर you could only separate fractionate proteins or any other materials either in the soluble portion or in the pellet form. So this is and then there was another centrifugation which was rate zonal centrifugation, in which you could make a gradient of density or mass. So all the proteins or all the materials which are present uh, in that assay, in that experiment, they will arrange themselves on the basis of their density gradient. Uh, you will not be able to separate any specific protein by either of these technique, but you will be minimizing the noise in a way that you have somehow put a window, a constraint on the basis of size or molecular weight. So all the rest of the proteins, they are gone. Then there is a second strategy which we use normally when we talk about proteins. It is through the electrophoresis, and this is done on the basis of charge to mass ratio. This is the characteristics which this technique is focusing. We have seen that the STS page, which is a polyacrylamide gel electrophoresis, could be used, and a pore size of this gel matrix is important because this decide uh, uh, what size of the protein you are going to separate from other protein, and we have seen that it is inversely proportional to the percentage of this polyacrylamide or whatever is the matrix इसके ऊपर ये सारा कुछ base कर रहा है. Before going into further, we have seen that before loading on the proteins onto the gel, you cover it with the STS page, you access the STS page. And because of that, all the proteins are denatured. There is no specific structure of the proteins available. So this is just simple linear structure now. So you will be going to separate them on the basis of molecular size. Smaller proteins will move quicker and they will be at the bottom of the gel, whereas the larger proteins and heavier proteins remained up. So you could be able by the end of the day to separate different fractions of the protein. So the largest protein, maybe medium sized protein, or the smallest protein. And of course, again, this is not the final thing. It might be that there are multiple proteins. For example, it could be 10-20 proteins which are of the same smaller size which are present here. So you would not be able to really pinpoint those proteins, but still you have again minimized the noise ratio, and you are now bit sure that your protein might be in one of those sections जिनके हम बात कर रहे हैं. Okay, so the proteins which are less than 10% of their weight, they are not uh, uh, they can be separated, but less than that you will not be able to separate them. Okay, there is a second technique. Uh, we are talking about the electrophoresis. There is another. This is one-dimensional electrophoresis. So think what we have done. We have separated molecules. We have separated proteins on the basis of their mass. So this is just one particular characteristic. So if you are lucky enough, you might be able to get your protein of interest, or maybe very small number of proteins present in that fraction, but normally do not happen. Normally we have multiple proteins of the same size or multiple proteins of the same ratio, so this is very hard. So it could be possible that you could separate proteins not only on the basis of mass but also on the basis of their charge. So this will be another technique which is called uh, two-dimensional gel electrophoresis. It's a very interesting and very important and very informative technique, and most of the time the people who are working, who are doing comparative proteomic studies, they use this technique. I would like you people to really pay some special attention what we are going to see in this technique. This is so powerful technique that you can even separate proteins as which have a difference in molecular size of 1 kilodalton. For example, a protein of 41 kilodalton, you could separate it from a protein of 42 kilodalton. This is a very, very powerful tool. So you would be interested into, so small differences can be separated in this through this technique. Uh, we have seen there is possibility that more than one protein could have this molecular weight. For example, this 41 kilodalton could be of 10-20 proteins within the cell, but it is theoretically it is not possible that all these proteins which have 41 kilodalton size, they might have the same sequence. So this sequence of these proteins might be different. If I am so it could be possible more than one protein do have the same molecular size, molecular weight, but all of these proteins theoretically are different, because we know that the composition of amino acids and the sequence of amino acids is different in each of these proteins. What does that mean? That means definitely then these proteins will have somehow a different charge. So that means this mass to charge ratio will be different in all these proteins which have these 41 kilodaltons, and you could take this as an opportunity to separate these all proteins. So how we normally do that, to separate these similar masses, the proteins which have similar masses, you could take another physical characteristics, and in this case it is the electric charge. So you could separate now the protein on the basis of electric charge as well as on the basis of mass. So that is why it is called two-dimensional. So one dimension is the mass, another dimension is the electric charge, and electric charge we know is determined by the amino acids which are present. So there are acidic amino acids, there are basic amino acids. So if there are more acidic amino acids, this protein might be positively charged. If there are more basic amino acids, this could be a negatively charged protein, and so forth and so forth. And we know how it is happening. So you could check, you could take this opportunity, you could take this information as an opportunity to separate this. So two unrelated proteins could be changed. So there is a depiction how you do it. So in one dimension you separate it on the basis of charge. So for example, negatively charged to positively charged, all proteins are separated, and then these separated proteins are layered up on SGS gel. मैं आपको अभी अगली स्लाइड में भी दिखाऊंगा. And then these proteins are separated just like before, जैसे हमने mass spec, जैसे हमने SGS page की बात की, on the basis of molecular weight, and now they will be separated on the basis of molecular weight. This technique is also called isoelectric focusing, because we have seen what is isoelectric focusing. Isoelectric focusing uh, based on the isoelectric points, and isoelectric points are the pH at which there is no net charge on a protein. You remember that? Though we have seen isoelectric point. So because in this technique we use this information, isoelectric focusing, so that is why it is also called isoelectric focusing. Okay, so this is how, this is just again a clear depiction and how a gel look like this. So you could first you separate the proteins on the basis of charge sorry, and then you resolve them on the basis of mass. So you have two techniques, you have two characteristics on the basis you are doing. So to separate on the basis of charge, you have a strip like this. So this is a commercially available synthetic strip made up of amphilites. I will talk about in the next slide again. what are these ampholites? So there are and can somebody would like to tell me what are ampholites? Anybody? एम्फोलाइट्स हमने पढ़े थे ना?
सर वो जो एज अ बेस और एज अ एसिड कन्वर्ट करते हैं
जो that carry both positive and negative charge. So these are called ampholites, and amino acids are in fact.
Yeah.
Okay, so this type of strip is commercially available, and which made it so you take a mixture of ampholites. So for example, positively majorly positively charged ampholites and negatively charged ampholites, and then you mix them, so this will create a gradient. So it might be that positively charged ampholites at one end and negatively charged ampholites at one end. So if you have an amino acid, if you have a protein loaded onto this column or onto this strip, so all the amino acids so all the proteins will migrate from top to bottom, and wherever there is an neutral pH. So you see, there is a pH at one end, and there is a pH different pH at the other end, and whatever the isoelectric point of a protein, it will stay there. For example, if a protein is basic in nature, it will be somewhere down here, and if a protein is acidic in nature, it will be somewhere up there. So any protein will adjust itself according to their isoelectric point. So this strip, which is made up of mixture of ampholites, and this creates a kind of pH gradient on this strip. So this pH gradient is at one end acidic, and at the other end is basic. So and all the if you load a protein onto this strip, so those proteins, when you will apply electric charge, so according to the electric, when you apply an electric charge on these protein, so these protein will migrate along with the strip. So wherever there is an isoelectric point, the pH where this protein have no charge, no net charge, so this protein will be staying at that point. For example, if a protein you loaded onto the gel has a neutral at a neutral pH remain is neutral, it will be here. So there might be proteins which are made up of acidic amino acids, more acidic amino acids, and these more acidic amino acids, because of these acidic amino acids, these proteins have an net positive charge. So if you load onto the gel, it might be moving to this end. So for example, it could be this point where this protein will be neutral, and if a protein is negatively charged and it has more basic amino acids, so it could be possible, uh, if you load onto the gel, this protein will be somehow neutralized at this point. So you see there are proteins, according to the compositions, these proteins will be neutralized at one isoelectric point or the other isoelectric point. Then what to do? So you first separate proteins on the basis of this charge, and now you see you see the whole principle what is going on here, and then you horizontally laid this strip. This is in horizontal. Pay special attention, what I am telling you is an important thing. So this strip, which now contain all the proteins arranged according to their isoelectric points, you load them onto the, not load them, you layered them, it's just the layering. So you have the gel, you have a gel something like this, and you layered that on the top of that gel. Of course, this pencil is in fact a small bigger one, so this gel will be of the same size as this wire. So what will happen? So now you will start. Now this strip will act as a kind of your well ये आपके वेल की शक्ल अख्तियार कर देंगे। जो प्रोटीन यहाँ पर मौजूद है वो एक किस्म का, जिस तरह हमने पुराना STS पेज में एक वेल के ऊपर प्रोटीन लोड्स की थी, वो तमाम की तमाम प्रोटीन अब यहाँ पर लोडेड है। सो व्हेन यू विल apply an electric charge again, so these protein will start moving into the gel, and now if for example this dot was made up of one protein only, it could be possible this has two proteins. Of course they have the same isoelectric charge, but it is hard that both of them could have the same molecular weight. It could be that this particular dot consists of more than two, for example, three proteins, and we know that it is possible that more than one proteins could have the same isoelectric point, but they will not have same same molecular weight, and same is the case otherwise. So it could be possible more than one protein can have one same molecular mass, but it is impossible or close to impossible that they have the same electric charge. So when you layer that strip onto the gel, so what will happen, all these proteins which are present in these dots will start resolving each other. If that protein is just one single protein, it will not further resolve. So for example, this was just one protein, it is not further resolved here. But this dot consists of three different proteins which have the same electric charge, but they have three different molecular weights. So there were some proteins which have much higher molecular weight, and then there were proteins which are smaller molecular weight, and there were proteins with the medium size, and so on and so on. So you see, so by the end of the day, your gel will be something like this. So it's a mess around. I mean, believe me, it's not that easy. It's not that beautiful as it is shown in this slide, but it will be something like this. So you will not have some dots, rather you will not have some band, specific band, but rather you will have some dots all spread on this gel. So just for a moment, please think for a moment what you see on this gel. I just want you to for a moment interpret this gel for me, and how you could do that. Think what are on the x-axis and what are on the y-axis. So can somebody tell me what is on the x-axis now?
Sir, isoelectric point.
Yes, isoelectric point. So that means this this x-axis is now, because this was the strip vertical, now it is horizontal, so everything is in the horizontal phase. So there is pH at one end, less pH at one end, so the pH is increasing in this direction. So that isoelectric points are increasing in this direction. So there are proteins with less isoelectric point, higher isoelectric point, higher isoelectric point, and the maximum isoelectric point. On the other hand, there are proteins which are arranged vertically on the basis of their, क्या है ये वाला? Mass.
Molecular weight.
Yes, molecular weight or mass. So you see, there are proteins at one corner with less molecular weight and less isoelectric point, are less positive, less charged. So we could say these are maybe the negatively charged proteins, if I'm not wrong. Uh, on the other corner, there are proteins with highest molecular weight and positively charged protein, if I'm not wrong, because it's just confusion sometime I create when I try to. So this if this is a negatively charged protein, then these are positively charged highest molecular weight protein. These are highest molecular weight protein present at this corner with maybe negatively charged fractions, because this we have said it's a less pH, so it seems it's a negatively charged. And then there are proteins with lower molecular weight, and then these are positively charged, and there are proteins which are in between them. So you see, majority of the proteins remained in the at the center. So every dot is different. So you want to know this protein, where my cursor is, I have just encircle this protein. So you want to know what are the characteristics. Okay, you go for diagonal, you go for horizontal, you have a particular information given there, so you can tell that this protein has a molecular weight maybe from uh, 35 kilodalton to maybe 43 kilodalton. So you cannot be sure what is exactly, but you can figure out, you can narrow it down, and you can tell, okay, this have maybe so electric point of roughly, maybe if it is 5.9, so it may be maybe 6. You just think of 6 and 6.1, 6.2. So you can say 5.5 to 5, 6.5. So you have an idea. So you can now guess what is that protein. So you have separated the protein in a much better fashion. Normally what we do when we do in laboratory such type of experiment, if we are interested in any of, we have any other information available, अगर हमारे पास कुछ मजीद information भी है इसके ऊपर, तो वो भी हम यूज़ करते हैं. फॉर एग्जांपल अगर हमें पता है हमारा जो प्रोटीन ऑफ इंटरेस्ट है वो इस मोलेकुलर वेट की है और उसका आइसोइलेक्ट्रिक पॉइंट ये है, तो हम इसको फॉर एग्जांपल इस डॉट को, सो यू कैन कट दिस डॉट आउट and then go for mass spectrometry in order to see what is it exactly, and so forth. And there are other tailor-made techniques which you could use in order to further identify this. So this is two-dimensional gel electrophoresis. Let me this is the whole philosophy which I have discussed in my previous slide, so you could see what is there. So how you do that, this is it's a simple two-dimensional gel electrophoresis. You take cell extract, first thing you took cell extract, and you denature it using urea, 8 molar. It's a high concentrated urea, and because of this high concentrated urea, this protein will be denatured. uh, so now uh, the this is ready for to load onto the pH gradient. As I told you, this is the pH gradient, and it is made up of mixture of amphilites, so poly anionic and polycatonic molecules, multiple polycatonic molecules and anionic molecules, and these amphilites are somehow mixed and made this gradient. So this is commercially available, commercially made gradient, so we might be sure that they are what they are claiming is okay. So that means if they are claiming that this end is the pH 4 and this end is the pH 10, they are fine with that. They know what they are saying. uh, so that means most acidic amphilites will be at one end and the most basic on the other end. So then you load your proteins on this strip, denature proteins, and these proteins will reach to their isoelectric point and they will arrange themselves. So this is called isoelectric focusing. So this technique is also called isoelectric focusing. On the other hand, when you talk about the second gel, when once you did it, you can go for second dimension, and this is on the basis of molecular weight. And for that purpose, now you put this strip into the SDS, it's saturated with the SDS. So now this protein is further denatured and all the secondary structures are gone, and now you could separate it on the basis of this. So you see, this technique is a powerful technique and it has been used quite frequently in order to do lots of experiments. And for example, if you are interested in differentiated, if you are interested in comparing proteomics of differentiated versus non-differentiated cells, or proteomics of cancers versus non-cancerous cells, you could use this technique. And so you have a differentiated cell and a non-differentiated cell, you crushed it, and then you go for whole of this procedure. You will separate them. So it's such a powerful technique. Simultaneously you could separate approximately 1,000 proteins. So this gel may contain approximately 1,000 proteins, and this is a huge number which could be separated uh, using this technique. So this is called two-dimensional gel electrophoresis. Now there is a third strategy. So we have talked until now two different strategies for protein purification. One was the centrifugation, other one was gel electrophoresis. Now there is a third uh, strategy which is called liquid chromatography. Can somebody tell me why it is liquid chromatography, not gas or any other chromatography? I mean, of course, there is a gas chromatography and liquid chromatography. Why only liquid chromatography?
So I think because we cannot extract protein in gas or that solid form.
Intelligent. Yes, because proteins cannot be present in the form of gas. Otherwise, uh, I mean, so that is why we are always talking about liquid chromatography when we talk about protein. So this is the liquid chromatography. It's a straightforward. Many of you know much better than me what is this technique, and of course, I believe you have learned this technique frequently in any other subject. So this is just a depiction how it is happening. So you have a sample, you load it onto the maybe any column, and by the end of the day, this column could be which is eluted. I mean, it's just a depiction. Don't think that it's always horizontal. So then by the end, at the end, there are different tubes. So these are different tubes, just and then there is a detector. So as soon as a molecule come to detector, it generate a peak, and we know what is that peak, and this peak has a particular time point at which this peak is appearing. So this time point is called its detention time. So we know at, for example, 3 seconds, after 3 seconds of elution, this peak will be appearing, and this peak will be might be the our peak of interest और हम इसी को ढूंढते कर रहे हैं. So we will majorly focus on this point then. Okay, so what is in general the chromatography originally so through the chromatography you could resolve proteins on the basis of mass, charge, and binding affinity. So these are three different characteristics which are given here. So we will try to focus only to these different characteristics because when we talk about proteins, so these are the major things जिनकी हम बात कर रहे हैं. So it could be able we could be able to use low molecular weight proteins from the high molecular weight. Initially, this technique was uh, developed for separating smaller molecules, for example, sugars, amino acids. But later on, you could use now this technique for protein purification and so forth. So what is how you do? So you prepared a sample and you put this sample on the top of a column, and it's a glass cylindrical column, and this column is filled with some matrix, filled with some material, and that material in fact determine what type of protein you are going to separate. What type of protein you are going to separate, this is going to uh, decide. So whatever the properties of these beads are, you will separate proteins. So you could separate proteins on the basis of mass. So you have a bead which could be able to separate on the basis of mass if you want to make, if you have a charge in your mind, so you could have a bead which could separate different charges, and if you have some specific affinity criteria in your mind, so these beads could fulfill this affinity criteria, and then you can go. So what is the principle? Molecules dissolve in a solution will interact. So principle is simple. So whatever the molecules you have, they will interact with these beads. So whatever these beads are, mass, charge, binding affinity, so these they will interact with them with this solid surface. So as soon as uh, you flush this column with some liquid, with some buffer, so they will slowly release them. So because now they are interacting with them, so there were molecules who are interacting less, and there are molecules who are interacting more. So those molecules who are interacting more, they will spend more time onto the column. So they will spend longer time to stay onto the column. अगर आप लोग थोड़ा सा देखें, ये पीक जो यहाँ पर नजर आ
सर
रही है, जी जी,
सर ये इंटरेक्शन किस बेस पे होगी सर?
मैं अभी उसकी तरफ हम आते हैं। इंटरेक्शन एनी बेस कोई भी हो सकती है, फॉर मैथ की बेस पे इंटरेक्शन हो सकती है, चार्ज की बेस पे, बाइंडिंग एफिनिटी की बेस पे, अभी हम उसकी तरफ आते हैं अभी वन बाय वन। सो this peak which is present at the end, or at the end, for example at this point I just came मैंने जिक्र कर रहा हूँ ये वाली peak, so this peak has the highest interaction with this column, so that is why it is coming at the end. On the other hand, this peak which is present in the beginning, they have the least interaction with this column, so that is why it is appearing quickly. So if if your proteins are interacting, they will spend more time onto the column, so the retention time will be more. If they are less interacting with the column, their retention time will be less. So this is the basic principle, and on this basic principle you can separate protein. So for example, if you want, मैं अभी हम मास की तरफ जाते हैं अब अगली वाली स्लाइड के अंदर. So if you are, okay, let's move on, so this will be easy to figure out. Okay, so this is, if you have a specific protein in your mind which have some specific molecular weight, uh, you could use it, and such type of chromatography is called gel filtration chromatography. So that means you are filtering out through a gel, and अभी किसी ने पूछा इंटरेक्शन किस किस्म की है, अभी मैं उन इंटरेक्शंस की तरफ ही आना हूँ. So how you normally do that? So you can make a gel filtration column made up of anything. So this is a, you what you have to make, you have to make a porous bead matrix. So that means any material which have some small pores in it, so this will be sufficient. By the way, our SGS page is a form of gel filtration. अगर आप लोग थोड़ा सा फोकस करें, क्या हमने किया था वहाँ पर भी, हमने पोर साइज की बुनियाद के ऊपर ही किया था. So this is the porous. So you can make this column through any material. For example, a polyacrylamide could be used, a dextran, which is a bacterial polysaccharide, could be used, and agarose could be used. Agarose is a seaweed derivative, so there is a seaweed from which this agarose is extracted, and you can make this column in there. So once your column is ready, you will load your protein onto the gel, onto this column. So there will be proteins with larger protein size, there will be proteins smaller. The yellow proteins are shown here, which are larger in size. Green dots are shown, which are smaller in size. What will happen? This is a porous column, which porous column. So the small proteins have much better chance to somehow bury, somehow trap in these small pores in a way that, for example, if this is a small surface, this small, if small, because small protein molecules could pass into that, they will be trapped in this hole or any other surface which is present, whereas the larger proteins, larger portions will remain on this surface. So they will remain on the surface, so they will not be trapped. So if you want to see what type of interaction is going on here, so this is a type of interaction, whereas where small size could be hold for longer period of time. So this is a type of interaction. When we say an interaction, that means uh, a measure of time which could be spent to separate this particular protein, a particular molecule from the gel. So this is a type of interaction in which uh, the size is the size is trapped in a big hole. So as a result, small molecules will not be easily escaping out of this pores, whereas the larger protein molecules will be separating out quickly. So because larger proteins are not interacting at all, so they are not trapped. So in this case, interacting means trapping. So large protein molecules are not trapped, so they are not interacted. So that is why they will be quickly coming out. So you will have a fraction where you have larger protein molecules. Then you will have a fraction where you have proteins with medium size. And then you have a protein fractions with small dots. So in order to release all these protein molecules, you are adding enough amount of buffer. So you use the buffer. The first run of buffers releases larger protein molecules. Second run are even more buffer, मीडियम साइज प्रोटीन, इवन मोर बफर, स्मॉलर साइज प्रोटीन. ऐसे जैसे आप हाथ धो रहे हैं, जितनी देर तक हाथ धोते रहेंगे उतनी देर तक का आपके वो जरासीम निकलते जाएंगे. थोड़ी देर तक धोएंगे तो मोटे मोटे उतर जाएंगे, थोड़ी देर तक ज़्यादा धोएंगे तो शायद थोड़े मजीद जो चिमटे हुए हैं वो भी उतर जाएंगे. थोड़ा और करेंगे, रफली फ्लश करेंगे पानी के साथ, तो एवरीथिंग विच इज़ ट्रैप इन ऑल दी पोर्स, जो कि आपको नज़र भी नहीं आ रहे, वहाँ से भी निकल आएगी. इट इज़ द सिमिलर. यू अगर आप थोड़ा सा अपने हाथ की या किसी जिस्म के हिस्से की तरफ देखें माइक्रोस्कोपिकली, देयर आर स्मॉल पोर्स, देयर लार्ज प्रोटीन्स. जैसा ये है, ये इसके साथ अटैच ही नहीं कर रहा तो इमीडिएटली नीचे गिर जाता है. स्मॉल डॉट्स कुड बी ट्रैप्ड हियर एंड दे आर नॉट गोइंग एनीवेयर. उनको निकालने के लिए आपको क्या करना पड़ा, अब इस पत्थर को उतारने के लिए आपको हाथ धोना नहीं पड़ेगा, ये खुद ही गिर जाएगा. लेकिन उस स्मॉल जर्म्स को उतारने के लिए आपको धोना पड़ेगा. एंड दिस इज द बफर. दिस सोप इज अ बफर एंड नाउ इट इज फ्लशिंग आउट एंड दिस इज. सो normally the volume depends on the volume of the column. So if you talk about small protein, small protein molecules, normally they require one column volume, minimum one column volume to elute. It could take more, but minimum one column volume is required for separation, because एक अगर कोई प्रोटीन मॉलिक्यूल, स्मॉल प्रोटीन मॉलिक्यूल इसके टॉप के ऊपर किसी जगह पर फंस गया है, तो उसको कम से कम इतना वॉल्यूम तो चाहिए होगा कि वो यहाँ से यहाँ तक पहुँच सके. सो दैट इज व्हाई व्हेन वी से मिनिमम कॉलम वॉल्यूम, दैट मीन्स इट इज देयर. सो I hope I have sufficiently uh, explained what interaction mean. Interaction mean a retention time. Any property which could hold the protein molecule is an interaction. In this case, a size is an interaction because it could hold the protein molecule, and this technique is called gel filtration. So if you have an idea of the size of the protein, you could go for gel filtration. If you do not have an idea of the size of the protein, you cannot go for the gel filtration. Okay? So normally we do load standards in order to go further. Okay, let's quickly move on to the second. I wanted to finish this gel chromatography today, because we will do further things next week. So second technique, which is called ion exchange chromatography, it's a simple, it is straightforward, the similar thing. Instead of having a gel matrix on the basis of mass, you are having a gel matrix on the basis of charged particle. So you could add positively charged beads, you could have negatively charged bead, you could say catonic chromatography, cation exchange for chromatography, you can say anion exchange chromatography. I haven't distinct both of them. मैंने सिर्फ इनको इनकी असल शक्ल में रखा है क्योंकि आप लोगों को असल पता है. So normally you can add amino group to the beads, you can add carboxyl group to the beads. So if you add amino group to the beads, so these will be positively charged beads, and that means negatively charged proteins could easily bound to them. If this is a negatively charged column, negatively charged, if you have labeled them with negatively charged particles, so this will be an cation, and so positively charged proteins will bind to that and they will interact with that. So longer the basic principle again, you want to increase the retention time. क्यों आप रिटेंशन टाइम इंक्रीज करना चाहते हैं? क्योंकि आप चाहते हैं कि तमाम गंद पहले निकल जाए, फिर मेरी प्रोटीन साफ सुथरी होकर निकले. Understand the point? So this is how we are doing that thing. So that is why we use a specific thing. By the way, मैं आप लोगों को एक छोटी सी चीज बताना भूल गया. Do not compare it with the gel electrophoresis. क्यों? क्योंकि जेल इलेक्ट्रोफोरेसिस में जो स्मॉल बैंड स्मॉल प्रोटीन मॉलिक्यूल्स हैं वो पहले निकलते हैं, लार्ज प्रोटीन मॉलिक्यूल्स बाद में निकलते हैं. ऐसे है ना? यहाँ पर उसके उलट है. यहाँ पर पहले लार्ज प्रोटीन मॉलिक्यूल्स एल्यूट होंगे, फिर स्मॉल होंगे. ओके, लेट्स कम बैक टू द आयन एक्सचेंज क्रोमेटोग्राफी. यहाँ पर यू हैव चार्ज बीट्स, पॉजिटिवली चार्ज, नेगेटिवली चार्ज. सो इफ यू लोड अ प्रोटीन quickly, if you load a protein, this, let's think this is a positively charged column. I do not have any specific meaning here given. So if this is a positively charged column, you loaded a protein which is negatively charged. So what will happen? All the positively charged proteins will not bound here, they will be repelled, so they will go away, so they will be eluted first. The negatively charged proteins will stay up on the column. And now you have to release those proteins by using again buffer. So you will add some salt, you will change the pH of the buffer, you could change maybe, of course the temperature do not play much role. It do play a role, but it might not be very successful way to do that. So you change the concentration of salt. So increasing charge might reduce this interaction of positively charged matrix and negatively charged protein. So slowly and gradually it will release the protein, and this protein will be separated out, and so forth and so forth. So you see there are different. So you could use positively charged column, you could choose negatively charged column. If you use positively charged column, it will be able to bind to the negatively charged protein. If you choose negatively charged column, it will be suitable to bind with the positively charged column. Okay, quickly, मैं बहुत ज़्यादा टाइम इस पे मज़ीद नहीं लूँगा, वही चीज़ें जो मैंने आप लोगों को अभी बताई हैं. So large, so you have, okay, मैंने फिर पीछे चला गया, क्या कर रहा हूँ मैं? इसका मतलब मुझे रोज़ा लगना शुरू हो गया. Okay, okay, okay. So this is the next. So this is the ion exchange. So net charge of the protein do play a vital role. So you choose. So at low salt concentration, protein molecules and beads are attracted by opposite forces. So that means बाइंडिंग के लिए आपने जो बफर यूज किया उसके अंदर साल्ट कंसंट्रेशन कम थी. सो पॉजिटिवली चार्ज कॉलम था तो नेगेटिवली चार्ज प्रोटीन बाइंड कर गई, नेगेटिवली चार्ज कॉलम था तो पॉजिटिवली चार्ज प्रोटीन बाइंड कर ली. बट एज सून एज यू चेंज द साल्ट कंसंट्रेशन, हायर साल्ट कंसंट्रेशन, दीज इंट्रैक्शंस जो कि पॉजिटिव और नेगेटिव थी अब ब्रेक होना शुरू हो गई. तो क्या होगा? पहले वीकली चार्ज प्रोटीन्स रिलीज होंगी, फिर हाईली चार्ज प्रोटीन्स रिलीज होगी एंड पे. सो जैसा हमने जेल फिल्ट्रेशन में भी देखा, अब जाहिर है पॉजिटिवली चार्ज कॉलम है तो नेगेटिवली चार्ज प्रोटीन्स बाइंड किए हैं इसके साथ. तो कुछ प्रोटीन्स के ऊपर -1 चार्ज हो सकता है, किसी के ऊपर -2 चार्ज हो सकता है. सॉरी, मैं गलत कर रहा हूँ. -2 चार्ज हो सकता है, किसी के ऊपर 3 हो सकता है. सो वट एवर देयर चार्ज, all negatively charged proteins are bound. So initially when you increase the salt concentration, those with -1 charge, those with -1 charge will release, and then those with -2 charge, then those with -3 charge, and then you have. So बेसिक आइडिया ऐसा नहीं होना चाहिए, मैं आपको बताना भूल गया, बल्कि आप लोग समझदार हैं खुद भी. इस सारी इतनी एफर्ट का ये नतीजा तो नहीं हो सकता ना कि बाद में सब चीज़ों को एक ही बर्तन में इकट्ठा कर लें. ऐसा तो नहीं हो सकता ना, आप एक ही बर्तन में इकट्ठा कर लें फिर तो फायदा ही कोई नहीं हो आपको सारी टेक्निक का. तो इसका मतलब यह है कि आप हर खास टाइम पॉइंट के बाद बर्तन तब्दील कर लेंगे. हर खास, मैंने कहा मैं 25 सेकंड के बाद ट्यूब चेंज कर लूंगा. सो 25 सेकंड में जितनी प्रोटीन्स आई वो इसमें आ गई, 25 से 50 सेकंड में जो प्रोटीन्स आई वो इस कंटेनर में आ गई, 50 से 75 सेकंड में जो प्रोटीन्स आई वो इस सेकंड में आ गई। 75 से कितना होता है 100 ठीक है। 100 सेकंड में जो प्रोटीन है वो इसमें आ गई। सो नाउ यू हैव डिफरेंट फ्रैक्शंस एंड व्हाट इज द डिफरेंस इन दीज फ्रैक्शंस? दीज हैव डिफरेंट टाइम पॉइंट्स। वो जो ग्राफ आपने शुरू में देखा, these different time points, not only different time points, because they are coming early, so they will be have proteins with negatively charged, they will be negatively charged proteins, क्योंकि हमने sorry they will be positively charged proteins, क्योंकि हमारा कॉलम पॉजिटिवली चार्ज है ना, तो ये बाउंड नहीं की, ये पहले निकल गई, ठीक है ना। मैं गलत कहूँ तो मुझे करेक्ट कर दीजिएगा किसी भी पॉइंट पे। these proteins have less negative charge, these proteins have more negative charge, मैं 2 नेगेटिव साइन लगा देता हूँ इसके ऊपर, and these proteins have even more negative charge. So you see, you separated different proteins fractions on the basis of charge now, and if you know what is the charge of your protein, for example, you know your protein has a net charge positive to negative to, so you will be more interested in this fraction, and I will take this fraction out and will go for further experimentation. I hope I have explained it sufficiently enough. So आप लोगों के लिए कोई और क्वेश्चन नहीं होगा। क्विकली एक फाइनल टेक्निक अफिनिटी क्रोमेटोग्राफी, बाकी मैं आज नहीं करवा सकूंगा आपको। सो द सिमिलर टेक्निक है, नाउ यू यूज दिस बाइंडिंग एफिनिटी। सो वो हमने देखा मैस की बेस पे आप सेपरेट कर सकते हैं, हमने देखा आप चार्ज की बेस पे सेपरेट कर सकते हैं। नाउ देयर इज द बाइंडिंग एफिनिटी। इट मींस इफ योर प्रोटीन ऑफ इंटरेस्ट हैज सम स्पेसिफिक कैरेक्टरिस्टिक to bind to a ligand, to bind to any ligand, you could use that ligand, you could use that ligand to bind to your protein of interest. So now your protein of interest is interacting with the column because it is specifically interacting, so not any other protein will be interacting with your column, so you have chance you will be able to separate this protein. So specific binding affinities of the protein to ligands are the basis of this affinity chromatography. So you could have a ligand molecule and you could use this ligand molecule. So ligand could be anything, small molecule, any antibody, any protein of interest, any substrate. अगर आपको पता है कि आपकी प्रोटीन फॉर एग्जांपल मैं कहता हूँ कैल्शियम बाउंड प्रोटीन है, तो मैं अपने कॉलम के साथ वो स्मॉल लिगेंड लगा सकता हूँ। ये देखो ना, आपको नजर आ रहे हैं ये लिगेंड, मैं आपको ये नज़र आ रहे हैं। अगर स्मॉल डॉट्स येलो डॉट्स के साथ स्मॉल रेड ब्लू स्मॉल डॉट्स, these are those ligands, you have those ligands bound to that column, so you could covalently bound those column with those ligand molecules, and you bound those column with ligand molecule. It could be an antibody, it could be a specific calcium molecule. अगर आपको लगता है कैल्शियम के साथ बाइंड करता है आपका कॉलम, या मैग्नीशियम में रहा नहीं अदर बॉल कॉलम, so you will have your protein loaded onto that. It's needless to say it will be neutral pH when you load this protein onto the gel, otherwise आपकी pH जो है वो इसकी बाइंडिंग को ही ख़राब कर देगी पकड़ के। अगर आपको पता है ना कि जैसे pH चेंज करेंगे तो प्रोटीन की स्ट्रक्चर चेंज होगी, प्रोटीन की स्ट्रक्चर चेंज होगी तो ज़ाहिर वो बाइंड नहीं करेगी, वो बाइंड नहीं करेगी तो आपका मकसद ख़त्म हो जाएगा। So that is why you go for neutral pH bounded and the protein will be bound and specific proteins will only stay there. So when you wash it, when you use buffer, all the proteins which are unbounded, they will be left, they will leave the column. Only those proteins which are your required proteins, they will be bound to this column. And now you elute those proteins using different buffer, different pH. So you could elute it 3 different methods. Quickly, these are excessive ligand. Either you could use more ligand to elute this. So आप फर्ज कर लीजिए आपकी प्रोटीन अब बाउंड है इस कॉलम के साथ और वो लिगेंड है फॉर एग्जांपल कैल्शियम। अब आप मजीद क्या कर सकते हैं, आप एक ऐसा बफर डाल लें जिसके अंदर कैल्शियम की भरमार हो, तो क्या होगा, अब इस प्रोटीन के लिए तो कैल्शियम की, पहले तो सिर्फ इस बीट के साथ कैल्शियम मौजूद था, अब कैल्शियम इस इन्वायरमेंट में मौजूद है, तो ये प्रोटीन उसके साथ बाइंड करेगी, तो इसका मतलब ये है now it lose its interaction with this column, and now you could separate it out. You can change the salt concentration. So changing the salt concentration again change the salt, change the pH as well as the interaction of protein with your column. So you could separate it. You could change the pH of the system. जैसा कि हमने pH 7 से start किया pH 3 पे, so you could separate it, and by the way by the end you will have different proteins जिनके अंदर different किस्म की characteristics होगी. So we have different proteins now separated. हम next class में further study करेंगे. कैसे मैं पीछे को क्यों बार बार जा रहा हूँ? आप लोगों के कुछ questions हैं तो मुझसे पूछ लीजिए ताकि मैं फिर जाऊँ. नेक्स्ट क्लास में हम एसेस की तरफ जाएंगे, फिर हम बात करेंगे. जी स्टूडेंट्स.
अस्सलाम वालेकुम सर.
जी वालेकुम सलाम.
सर असाइनमेंट के लिए एक दिन दे दें.
आला, मैंने क्वेश्चन क्या पूछा, आपने दिन पूछ लिया. भाई सबने करवा दिया, फजल क्यों पीछे रह जाते हो हमेशा?
सर वो डाउनलोड नहीं हो रही थी, मैंने आपको बताया था नेट की वजह से, फिर रिजवान से मंगवाई थी मैंने.
अच्छा चले करवा दीजिए, कल तक करवा दीजिए, ठीक है
सर वो ऑप्शन ही नहीं आएगा तो
मैं कर दूंगा उसको ओपन कर.
ठीक है सर, थैंक यू सर.
और कोई?
और सर हमने वो क्विज मिस किया था सर, आपसे बात भी हुई थी, आपने कहा था कि बाद में अरेंज कर लेंगे सर.
हम करते हैं, 2 बंदे थे ना आप लोग, कौन था दूसरा कौन?
सर दूसरे का तो पता नहीं सर.
मेरे पास वन है. ओके चले हम देख लेते हैं, इंशाल्लाह करते हैं इसको.
ठीक है सर.
ठीक है
ओके सर.
ठीक है. और कोई क्वेश्चंस हैं किसी के? नो क्वेश्चंस, then we will leave and inshallah we will meet next week. ठीक है. ओके जी, अल्लाह हाफिज.
अल्लाह हाफिज.
ओके सर, अल्लाह हाफिज.